Like everyone had your physical. All right, so today we're gonna finish up with our arterioles and dive into capillaries. So we're gonna start with a rapid fire little set of review questions on our arterioles. You don't have to put a real long name in here. Your initials will be fine, but I think it will ask you for a name if you join our little quiz. It's not a quiz, I'm grading. It just lets me put in more questions. No. No, none of my friends. Well, like, we didn't discuss, I think last year, like, this is a nursing program now, it's usually what we do. So I think actually we can get that to. Because the first thing is, Alan, that's all stays up here. Exactly. Sorry, no, I forgot. Oh, that's okay. I was just wondering. We can do the ball club on Sundays. We can. We can just go. You know, yeah. Yeah, I forgot. No, I was not, yeah. I'm down. Yeah. All right, everybody ready for some review questions? Yes, awesome. Okay. So these are about the topics we were talking about last time with our extrinsic control of arteriolar resistance. Okay. All right, let's take a look. Okay, perfect. Yes, the answer is the sympathetic nervous system. Okay, so sorry, everybody got nervous in the order. Sure. Okay, yeah, so our efferent nerves going out are motor nerves. One of our branches is the autonomic nervous system. We have sympathetics and parasympathetics. It's those sympathetics, the fight or flight part of the nervous system that we were talking about last time with all those different adrenergic receptors that we see in the smooth muscle in the walls of our arterioles. All right, for the next question, you're talking about those receptors. So which was the one that caused vasodilation? Right, we're gonna divide here, but it is the beta 2 receptors. Okay. So we did talk about the alpha receptors as well. Those were the ones responsible for vasoconstriction. Both types of adrenergic receptors, they have different functions. So where were we looking for those beta 2 receptors? Yes, beta 2 receptors, specifically those beta 2 adrenergic receptors, are found in the vasculature going to the skeletal muscle and the cardiac muscle. So the beta 2 cause vasodilation. Which receptor is responsible for vasoconstriction? It can go both ways. Bigger vessel, smaller vessel. Okay. So you were listening when we went over that last question. Okay, so beta 2 receptors were the ones doing vasodilation. The alpha receptors are the ones responsible for vasoconstriction. So next, I'm going to ask you the same question as before. We're going to look if you want to find an alpha receptor and only an alpha receptor. So arterioles throughout the body, except the brain, have alpha receptors. Okay. So the reason I specified only is that your arterioles going to skeletal and cardiac muscle do also have alpha receptors, but they also have those beta 2 receptors that got both. Everywhere else in the body, we've got these alpha receptors, but not in the brain because it's doing its own thing. And this is why we see a big effect on total peripheral resistance, right? Total peripheral, so everywhere out in the body, when we activate our sympathetic nervous system, partially is going to be because of those alpha receptors throughout the body. When we think about distribution of blood, when our sympathetic nervous system is activated, what's going on with that blood? Where is it going? Are going more to muscles or heart or less to muscles and heart? Okay, great. Yeah, so that's the big takeaway here, right? Like that's what all of this is building to, right? So alpha receptors out most places in the body cause vasoconstriction when norepinephrine coming from the sympathetic branch of the autonomic nervous system binds to them, right? So they're raising resistance. So most places in the body, because we're raising a resistance, are kind of lowering the proportion of flow that they're going to get to them. But because we also have those beta 2 receptors in the arterioles going to the heart and going to your skeletal muscle, especially at the beginning when we have low concentrations of epinephrine kind of building up, right? They're getting that vasodilation effect, increasing the flow, going to those parts of the body, preparing you to have your heartbeat harder and faster, letting you run away, because the sympathetic nervous system is all about that fight or flight response. So that's what's going on as we're doing this extrinsic control of the arterioles and therefore blood flow and resistance with our sympathetic nervous system. How about you got 6 out of 6? Okay, I'm not going to embarrass anybody. All right. So this is part of how we were doing our regulation of blood flow. And especially during exercise, we think about it because our cardiac output is increasing during exercise. We know cardiac output is what we're subbing in for flow in that flow equation, right? So cardiac output is going to be equivalent to our mean arterial pressure divided by our total peripheral resistance. The overall effect of the fact that we have this vasodilation when we have, especially epinephrine binding to those beta-2 receptors, headed to the heart, headed to the skeletal muscle, is that we have more of that flow going to those places. We have alpha receptors everywhere else. So places like your gastrointestinal tract, places like your kidney, you're going to get less blood flow going to them. So, you know, hopefully while you're running away, you don't pee yourself, right? Basically. And you maintain your blood pressure, right? Because otherwise vasodilation, if it was happening overall, dropping your blood pressure too low would make you feel kind of faint, right? So that's also going to be important for maintaining your function under these conditions of stress. Okay. So questions about this topic? So there are some other factors which we would consider extrinsic control on our arteriolar radius and therefore resistance, and therefore also our mean arterial pressure or blood pressure, because all of these things are kind of in relation to each other. Okay. So epinephrine should be no surprise. That's coming from the adrenal medulla, and we just saw that that's really part of the sympathetic response, binding to those beta-2 receptors, right? So we know that's causing vasodilation, at least at those first low concentrations. As it rises, spillover gets to the alpha receptors. Okay. The other 2 hormones that you may hear about relating to our arteriolar radius, our blood pressure, are vasopressin, also known as ADH, antidiuretic hormone, which is how we'll see it referred to when we get to the kidneys, because it's going to be really important in our renal unit. But we've seen it before, briefly mentioned. It's one of our hormones coming from the posterior pituitary in our endocrine unit. Usually what we think of its job as is increasing water reabsorption by the kidneys. So it's going to be pulling more water back into your body. It's going to have the effect of basically like keeping your blood volume up. At the same time, its effect on arterial radius is vasoconstriction. Okay, so it's keeping your blood pressure up in kind of 2 ways, we'll see. Another hormone that has effects here is angiotensin 2. This increases your total peripheral resistance. It does some vasoconstriction. So these you might see referred to in different kinds of conditions, different blood pressure issues. So next we're going to move on to our capillaries. So, thus far, right, big picture, we talked about the heart. We know blood's coming out of the heart. It's getting pumped into the aorta, just swelling, holding pressure in its walls. And then that aorta is continuous pressure throughout our systemic circulation. We're going through big arteries first, and they get smaller. Then we have our arterioles. We think a lot about that smooth muscle in the arteriolar walls, because this is where we control their radius and therefore resistance throughout our system. So the next place we're going with this blood, after we've determined how much flow we want to different regions, So we're going to the capillaries, where we're going to be actually exchanging stuff across the walls of the capillaries in order to basically feed our tissue and flush out our waste products. So we're going to take a brief look at capillary anatomy, so what like a capillary looks like. And we're going to briefly touch on how we control blood flow through those capillary beds. And our big topic that I recommend you focus on as you're studying is how and why that movement happens across the capillary walls. And that's a real purpose of our capillaries, is to allow that transfer. And so that's our big takeaway when we get there. So capillaries are our site of exchange. They're small, which means we have a nice small distance for things to diffuse in and out of the plasma, in and out of the blood, into the interstitial fluid, and then into our tissue. Now, capillary, its wall is just one cell layer thick, basically. So we're just going to have like a layer of endothelial cells, just like one little layer, right? Because we want stuff to be able to get across. We're going to want to control and predict what gets across, but we do want things to be able to cross. Our capillaries are going to have our greatest total cross-sectional area in our body, which really just means you've got lots of capillaries, right? So they're not very big, but you have tons of them. You have tons of capillary beds everywhere. And because there's so much areas, I think of it as like room for the blood to kind of spread out through all these tiny branches. We also have slow blood flow, as that flow is kind of like diverting through all of those capillaries, which means that we also have kind of time for things to cross our capillaries. So this is all helpful for us as we're trying to get nutrients, gases crossing our capillary walls so that we can maintain our body functions. So they're up to like 40 billion of them in your body. Their total surface area is 600 meters square. Almost all your cells are within a single millimeter of a capillary. So they're really distributing your blood flow as close to all your cells as possible. They're also usually only like a millimeter long. They're small. I drew those little yellow circles with some gaps between them because your capillaries do have pores. So what I was drawing was that inner endothelial cell layer. So kind of like your epithelium was the outside layer of something. And the endothelial cells are lining the internal part of our blood vessels. Okay. The pores are big enough for some things to flow through, but not others. Basically, everything but the proteins in your plasma, in your blood, are going to be able to travel through capillaries, depending on their type of capillary. Okay. Anything bigger than a protein is also not going to go through. So like, you're not going to end up with like red blood cells escaping, ideally, through a capillary. When we refer to a capillary bed, all we mean is that we're talking about a big network of capillaries as opposed to one little individual. Okay. So if we look at that area, what this is emphasizing to you is that because we have so many capillaries, right, because our blood flow is basically branching over and over again, right? We'd have an artery going to a bunch of arterioles, each going to a bunch of capillaries, right? This is how we end up with that big surface area, right? Because we have this whole branching system. Okay. So we've got a lot of cross-sectional area when we add together all our capillaries. And the speed, the mean velocity of the blood going through our blood vessels really corresponds to how much space they're spreading out through, right? So as we end up with more and more space, more and more area, we see a drop in the velocity of the blood going through those blood vessels, which is why we have such slow flow through the capillaries, which we love. Okay. So we do have two types of capillaries that we want to think about. Most of the capillaries we talk about are continuous capillaries. So continuous capillary, it's going to just have small gaps between those endothelial cells, right? So they're going to allow some small water soluble, so hydrophilic molecules to go through their pores, but they're not going to allow big things to go through, right? So we've got relatively small gaps. So we're going to think things like ions, basically, right? Fenestrated capillaries. Fenestra means window in Latin. So they're capillaries with big windows. So they have large gaps between those cells, okay? So if you are in a region of the body that has a fenestrated capillary, this is where you would have some big things flowing through. Fenestrated capillaries are rare, but if you have one, you might let proteins, might even let some blood cells to go through in those regions. So unless I say otherwise, we will be talking about continuous capillaries. I'm not sure we come back to fenestrated capillaries at all, but I'll flag that if we do. Okay. There are other forms of capillaries that we are going to completely ignore, called discontinuous capillaries and sinusoids, which are specific to specific regions of the body. Okay. We're just going to go with our generic continuous capillary. Okay. So we're going to take a look at how we have the microcirculation, so our arterioles, our capillaries, our venules, working together, working with the smooth muscle in there to kind of control how much blood goes through a capillary bed. So here we have a picture, okay, that has a structure called a meta-arteriole in it. So what we're looking at here, right, our arteriole is the part of the microcirculation that has blood coming from the heart, right? So here's our arteriole, okay. Going back to the heart, we have our venule. The capillary bed is this network in between, okay. And what a meta-arteriole is basically a shunt. So like a big bypass on a highway, right? This is our meta-arteriole, okay. So basically what a meta-arteriole does is, if you can get it, if you are a little bit of blood, right, and a meta-arteriole is open, right, you can go through that meta-arteriole and kind of skip the capillary bed, right? So if we open and close these meta-arterioles, we're going to be able to either divert blood into the capillary bed, or it's just going to speed along way past and kind of skip over the capillary bed due to that low resistance in that meta-arteriole because of that larger size. Okay. Now, in order to open and close these areas, we have rings of smooth muscle. So we can see, for example, a precapillary sphincter here. So we got some smooth muscle here. And we can see those rings of smooth muscle that we've been talking about wrapped around our arterioles. We can see that we have some smooth muscle wrapped around the meta-arteriole itself. Okay, so these bits of smooth muscle are going to be contracting and relaxing in response to local factors. So think kind of like when we were talking about the effects of oxygen and carbon dioxide on the arteriolar smooth muscle, that sort of thing. Okay. When they contract, they're going to end up increasing blood flow through the capillaries. When they relax, they're going to decrease blood flow through the capillaries. So they're basically either cutting off the meta-arteriole or relaxing, letting blood go through the meta-arteriole and therefore skip over the capillary. So we can do some fancy control of specific capillary beds through the action of these meta-arterioles with their smooth muscle. The precapillary sphincters specifically, right? So the meta-arteriole, right? So I should point out, right? So the meta-arteriole, right? You can think if it gets narrower, right? It's contracting, it gets narrower, right? Its resistance is going up. So its flow is going down, which means that blood has to go somewhere, it goes through the capillary. Right, so that's what we were talking about on this slide, which is different than what we're talking about on this side with the precapillary sphincters. So we're still doing intrinsic control here, just local factors, okay? But what we're doing is cutting off this capillary or not. Okay, so if this part shrinks, right, if we constrict that entrance to the capillary, this is going to decrease the blood flow, right? Because we're kind of sealing it off. If it relaxes, opens, can increase the blood flow here. So when we say metabolites are causing this relaxation, right, think like carbon dioxide and stuff like that. Just local factors. Okay. Getting to our big topic. Okay. So what we care about most about capillaries is that we can feed the tissue, we can get stuff moving towards the tissue. And whether we like it or not, we're going to have movement of fluid across these walls of the capillaries. Okay. So when we are seeing materials across our capillaries, the most common thing that we're going to see is we're going to see diffusion. Okay, so diffusion, remember, is one of our types of basically passive transport, right? So we don't need energy here, but we might need a channel, okay? Depending what type of molecule we have, right? Because if we have something that's lipophilic, meaning it likes lipids, right? Then it can go through that endothelial layer, right? It's going to go through the cell itself. Okay. So something that's lipophilic can just cross straight through because it can get through that lipid bilayer. Something that's lipophobic is going to have to go through a channel, right? We're going to have to make a little door for it. Okay. What we're going to see going through the pores is mostly going to be like water and things that are dissolved in the water. We also have other ways that stuff can cross in specific situations. So transcytosis is when we have something go across by having bits of the membrane pinch off and then let us release pockets of stuff. So think about kind of how we release neurotransmitters in synapses, right? That's kind of what we're thinking about with transcytosis. So some proteins cross in that manner. And in the brain, we have mediated transport, so it's going to be controlled by things like our glial cells. Okay. So I asked you to look over, at least in brief, some of these topics before we start in class today. So we're going to be talking about filtration and absorption and osmotic and hydrostatic pressures. So I want you to do the best you can to tell me which of these is a description of filtration. Take another couple of seconds. Okay. Okay, so when we're talking about this next topic, we're going to be focusing on filtration and absorption. So what we're talking about, think like filtering the blood. We're going to be thinking about movement from the blood into the space around the capillary, so into that interstitial fluid, which is a type of extracellular fluid. And the opposite is going to be our absorption, so coming from that region into the blood. Right. So when we're thinking about the materials that are moving across the walls, one of the most important things we're tracking is fluid traveling across that wall, because that's also carrying the stuff that's dissolved in it. Okay. So what we have moving, the fluid we're talking about is plasma minus the proteins. Okay. And we want to know our directions first. So filtration means out of the capillary. Okay. And then absorption means into the capillary. And what we're going to be doing as we start to calculate our net filtration pressures and think about this movement of fluid across our capillary walls is really what we're trying to figure out is, do we have more forces forcing blood out of it, forcing fluid out of the capillary? Do we have more filtration happening? Or do we have more forces pulling fluid into the capillary? Do we have more absorption happening? So we're going to have forces responsible for both of these. Okay. And our job is going to be to figure out which one's winning out and by how much. Okay. Now, the purpose here is to help us distribute extracellular fluid, maintain those levels of solutes, basically allow stuff to flow through tissue, right? So plasma was one of our types of extracellular fluid. And then our interstitial fluid was another type of extracellular fluid, right? So by allowing fluid movement between these two, we're going to help keep those distributions as they should be. Okay. So here we're looking at that capillary cross-section. So we're remembering what can go through, what can't, and therefore what's going to be in that plasma that we are filtering or potentially absorbing, right? So here are those endothelial cells. We've got some pores at the corners, so we can see those pores here, okay? So we have things that can dissolve in water-soluble substances able to travel through those pores, right? So we're going to expect these things to be moving along with our fluids, right? So this is our salts, our sodium, our potassium, our glucose can move as they choose. Lipophilic things can go directly across our cell membrane. So like oxygen and carbon dioxide are actually just diffusing along their own gradients so that they don't need this fluid movement. Proteins we'd have to control going through. All right. So the other thing I asked you to take a look at was the difference between osmotic pressure and hydrostatic pressure. So take a second to pick which of these you think best describes what osmotic pressure is. Okay, take another couple seconds to pick an answer. Let's take a look. Okay. So what we are actually talking about, oops. Sorry, I picked the wrong one. I just autofilled A. When we're talking about osmotic pressure, what we are talking about is the solutes. Okay. So we're talking about two pressures, osmotic and hydrostatic. Okay. So osmotic is like osmosis, right? So when we talked about osmosis, we talked about how when we have lots of particles in some region of fluid, right? If those particles can't cross our cell wall, right? If they're non-permeable, non-permeate, impermeate, this means that water is going to move towards them in order to try to like water them down. Okay, so that's what our osmotic pressure is going to be. The other one is going to refer to our hydrostatic pressure. Oh, maybe I was just picking the top one. Anyway, our hydrostatic pressure is going to have to do with essentially columns of water, like exerting a force pushing away. All right. So when we're talking about these two forces, we can think of them as like pushes and pulls. Okay. And we're going to see a lot of abbreviations. So let's start that out now. Okay. So when we are talking about our osmotic pressures, we're going to see a little pi. And when we're talking about our hydrostatic pressures, we're going to see a big P. Okay. So the hydrostatic pressures, I think of, what do you think of it like normal pressure, like pressure against a wall, right? So if we're thinking about our capillary, right, the blood in that capillary is going to push against the wall of the capillary with some strength, right? So that's going to be one of our hydrostatic pressures. That's going to be the hydrostatic pressure of the capillary. Any fluid that's building up outside of the capillary is also going to exert a push. It's not usually going to be a super forceful push, but if you think about like swelling, right? When your skin gets like all tight when it's really swollen, right? You can think about how that fluid is pushing, right? So you'd have a push back in, that would be the hydrostatic pressure of this space around the capillary, of this interstitial space. Okay. For our osmotic pressures, right, we have something different going on. What we're talking about with our osmotic pressures is we have a pull from those solutes, right? So we're leaving a lot of proteins in our plasma, right? Because they can't escape, they can't go across that endothelial cell layer. So we're going to see that those proteins in the plasma exert a pull towards themselves, right? Because any water out here is going to try to come in and water down those proteins that we're leaving. Okay, so that would be the osmotic pressure of the capillary, because that's where these proteins are. And if we were to have anything dissolved here in our interstitial fluid, right, those solutes would also exert a pull. That pull would just be towards themselves. So the osmotic pressure of that interstitial fluid would be out of the capillary. So these are the four things that we're really considering as we think about, okay, overall, are we going to have filtration or are we going to have absorption happening in this capillary, in this place at this moment? Okay. So sometimes you will see the osmotic pressure when we're talking about capillaries referred to as oncotic pressure. The difference there is that our only non-permeating solute that we're thinking of is proteins. So oncotic pressure is just being specific, that we're talking about that pull from proteins specifically, whereas osmotic pressure is not specifying that it's proteins, it's just more general that we're talking about any non-permeating solute. Okay, so for our purposes, we can think of osmotic and oncotic as kind of the same thing. Okay. So we're going to start with that hydrostatic pressure. So both our interstitial fluid and our plasma in the capillary have a hydrostatic pressure, right? Okay. Because they both have fluid in them, right? I'm going to draw a water droplet in each, right, for our fluid. Okay, this is what we're talking about, fluid. Okay. So the capillary hydrostatic pressure, which we'll see abbreviated ECAP. What's creating this pressure is essentially the blood pressure, right? So the blood pressure that is in that capillary, right, is pushing out on the walls of the capillary. That is our capillary hydrostatic pressure. So it's pushing out towards our interstitial fluid. Okay. So based on that direction of the arrow, right, I really like to draw while we're doing these things because it helps me remember what force goes with what. So remember, we had filtration meant going out of the capillary. We had absorption going into the capillary. All right. So we can see that the arrow that was drawn, that blood pressure in the capillary pushing out, matches our arrow for filtration. And indeed, the hydrostatic pressure of the capillary is a force that favors filtration. Okay? So if you have a high blood pressure coming into that capillary, you have a big force that's going to lead to filtration if it can overcome the other three forces that we're going to look at. Okay. At the arteriolar end of a capillary, right? So we have our arteries on one side, arterioles, and we have the venules on the other side. So we're talking about like here. So we do have a difference in the pressure, right? We have a delta P, a difference in pressure across the capillary, which is why we can have flow through the capillary, right? We need a pressure difference in order for flow to exist, okay? So at the arterial end, we're gonna have a pressure PCAP of 38, okay? But as we lose fluid, because we are gonna see that at least at the arteriolar end, right? We're going to have, on that, we're usually gonna have filtration. So we are going to see that pressure dropping as we move forward. So as we get to the venule end, we have a lower pressure, a lower hydrostatic pressure in that capillary. So this is a force that changes depending on rate. But it still does have a positive pressure, right? Just not as big as like the arteriolar end. Okay. Now, we also have our forces from the, okay, from the fluid in that interstitial space, okay? So this fluid, likewise, is exerting pressure from where it is. So it's pushing back towards the capillary, right? So that's our pressure of the interstitial fluid of that interstitial space. Specifically, it's hydrostatic pressure, but it's like almost zero. There shouldn't be a lot of fluid buildup around the capillaries under normal conditions. But that direction, right, does match our absorption or reabsorption here. Okay, so if we had that number get really big, we'd be favoring absorption, so favoring movement back into the capillary. So if we're looking at these forces of our just our hydrostatic pressures, we're going to see at the arteriolar end, we had the pressure in the capillary, the hydrostatic pressure in the capillary was 38. That favored filtration. That force fighting against it coming from that interstitial space, from that interstitial fluid. We're going to subtract it out because this is like a tug of war situation. We've only got the fluid that we're trying to move. So we subtract that force pushing back into the capillary for 37 millimeters of mercury as our force for filtration from the hydrostatic pressures. At the venous end, on the venule side, we have a lower hydrostatic pressure in the capillary, but we still have barely any hydrostatic pressure coming from that interstitial fluid. So we still have a force for filtration, it's just less. So those are hydrostatic pressures. Next, we're thinking about our osmotic pressures or our oncotic pressures, because we know that this is mainly from proteins. Okay, so I'm gonna redraw my little filtration absorption cheat sheet, right? So filtration is movement out. Absorption or reabsorption is movement back in. Okay. So as we draw these on, we're just going to match up what these forces are. Okay. So when we have a capillary with a bunch of proteins in it that can't get across that wall, they are pulling on that fluid to come water it down, right? Specifically, we have a pull, an oncotic osmotic pressure in the capillary, right, of 25 millimeters of mercury. Okay. So we can see, based on the direction of that arrow, right, this is a force for absorption or reabsorption. And we're using that terminology because we will also be talking about these forces in the kidney. And there are going to be some subtle additions when we get there. Okay, but for now, absorption, reabsorption, ignore the difference. Okay, cool. All right, so that's our direction for the osmotic pressure in the capillary. In that interstitial fluid, we have very little dissolved impermeant materials, but they do exert theoretically a pull towards themselves. Okay, we can see that's a 0 to 1 millimeters of mercury. But we can see if they did do much pulling, that would be a force for filtration. All right, so we can see that's the other direction. Okay, so the pull of the interstitial fluid. Okay, cool. So in terms of these osmotic pressures, we have proteins in the capillary, so pi cap of 25, pretty much nothing coming from that interstitial fluid, unless we have something weird going on. So we have overall a force for reabsorption when we're looking at those osmotic pressures. Right. So we are going to tie this all together. Realize it is the end of class, so we will save practicing these for next time. But what we're doing, right, is basically we're going to be taking all of our pressures for filtration and summing them together and subtracting out our pressures for absorption. Okay, so if you have not looked at this section on net filtration pressures in your textbook, recommend you do so before Wednesday so that we can keep talking about this and make sure it makes sense.